Good morning, everyone, and welcome to today's webinar. My name is Alba Flores. I'm Marketing Director at Reference Laboratory Genetics, and it's my pleasure to introduce you to our colleague and Scientific Director, Dr. Manuel Martínez.
Morning.
So let's start with the presentation. Before beginning, please allow me to briefly introduce you to our laboratory. Reference Laboratory was founded in 1975, over 45 years ago, and today is established as one of the main European laboratories in spatial analysis, collaborating with more than 400 hospitals and over 1,000 laboratories all over the world. Reference Laboratory Genetics is our division of molecular and genetic diagnostics. For those of you who already know us, you are aware that our patient is developing new diagnostic tools that will have a significant impact on the quality of life of our patients and their families. We look at each case individually and recommend the most appropriate genetic diagnostic studies, always prioritizing the well-being of the patient. We have divided today's session into 6 sections. Introduction to the non-invasive prenatal testing, why your screening should not be limited to common trisomies, VeriRef platform, limitations of the technique, clinical cases, and finally, we will finish with some conclusions. Oh, and before I forget, at the end, we will leave a few minutes for questions and discussion. I'm sure that you will have some interesting questions to share with the other attendees. Remember that during the session, all attendees will be muted, so if you have any questions, you can use the chat. So if you are ready, we can begin with a short introduction to the non-invasive prenatal testing. Nowadays, there are many different screening and diagnostic options available for the detection of chromosomal aneuploidies, and most of them depend on the stage of gestation. The traditional screening options are shown in blue and include the combined screening from the first and second trimester. The main disadvantages of these traditional screening methods are the following. Even when carrying out a complete screening through the whole pregnancy, there is a high rate of false positives, around 5%, and a detection rate of only 60–90%. Also, they only allow us to screen a small number of chromosomal anomalies, like trisomy 21, 18, and in some cases, trisomy 13 or the monosomy of the X chromosome. They can only be carried out for a short time during pregnancy, and as you can see, during the third trimester, there doesn't tend to be any screening method available. If traditional screening indicates that the fetus has a high risk of aneuploidies, or if the patient has other risk factors that justify it, prenatal invasive testing is currently the only clinically available method to definitely diagnose a chromosomal alteration in the fetus, either from chorionic villi or amniocentesis. Cytogenetic studies are carried out in order to obtain the fetal karyotype. The problem with both invasive procedures is that they carry a certain risk of fetal loss. With the introduction of the non-invasive prenatal testing, NIPT, as a screening method, it's possible to reduce false positive of traditional screening. As you see, from 5%, we passed to a less than 0.1%. Therefore, it's possible to reduce confirmative invasive procedures and the associated fatal risk. What's more, it shows over 99% sensitivity, which allows a higher detection rate. Importantly, as well as being a very easy test that only requires a sample of maternal blood, it can be carried out from week 10 right up to the end of pregnancy. Oh, and of course, it detects other sorts of chromosomal alterations that are not included in traditional screening. What we must make clear is that not all available NIPT technologies offer the same possibilities. Some of them have a higher test failure rate and show lower sensitivity, especially in low fetal fractions. It could also be that they are not suitable for some one-current twins, egg donors, patients with high body mass index, or for certain ethnicities. What's more, they could have shorter analysis windows or not be CE marked or only be CE marked for trisomy 21 or certain parts of the test. And most importantly, most of the technologies on the market tend to be limited to the most common trisomies. In more detail, and in contrast to the aforementioned technologies, VeriRef Gold, our non-invasive prenatal testing, is a highly accurate test that detects the risk of aneuploidies and copy number variations in all chromosomes, studying the fetal DNA in the mother's blood. It also provides information on fetal gender. VeriRef Gold is carried out in Spain on the premises of reference laboratory genetics, thanks to a technology transfer from Illumina, the world leader in NIPT technology. 4 important points that back up our test are that over 1 million samples have been analyzed, that it is the test with the larger number of publications that back it up. We will share some of these publications with you through this presentation. And in addition, the whole test is branded as CE EPG, and it also has the technical accreditation 15189. All this data and the certifications allow us to ensure that it is the test with the lowest rate of non-obtaining results, the lowest false positive rates, and the highest sensitivity and specificity. In addition, the optimization of our procedures and the high number of samples that we process enable us to offer a very short turnaround time, between 3 and 5 working days. What's more, in order to ensure that we offer the best service to all healthcare professionals, when a high-risk result is obtained, we immediately inform to the test prescriber and offer free confirmation via QF-PCR or CGH array. We also offer pre- and post-study genetic counseling with Dr. Manuel Martínez and Dr. Cristina Camrubí, specialist in reproductive genetics. And of course, integration of the results is possible with any laboratory computer system. All this from week 10 of gestation and for all types of pregnancies. Now let's focus on the question, why your screening should not be limited to common trisomies. Well, we know that fetal chromosomal anomalies aren't limited to the most common trisomies. In this graphic, you can see the results from the study published by Diana Wesley in 2012. Dr. Wesley determined the frequency of various chromosomal anomalies. As you can see, rare chromosomal anomalies, these 60% here, are as common as trisomy 18 and trisomy 13 together, or they are more frequent than sexual chromosomal aneuploidies. It's clear that individually they are not common, but collectively they are significant. Unfortunately, rare chromosomal anomalies are not included in all NIPT technologies. It's possible that the justification given by some NIPT providers that don't include rare chromosomal anomalies is that including them, the unreliability of the test would increase massively, when the most likely reason is that they are not included because of technical limitations. In fact, focusing on bibliographical data, like this article from Fertile, which includes around 90,000 pregnancies, we can see that with expanded NIPT, the positivity rate of the test is only increased by 0.44% in the population of average risk. In fact, most of these positive will be real positive and will have a clinical impact. This table simply shows in blue positive rate for standard NIPT with 0.56%, in red The incidence of positive when sexual chromosomes are analyzed with 0.39%, and in green, the incidence of positive when the expanded NIPT is carried out, with an increase in positives of just 0.44%. Another reason to not restrict your screening to common trisomies is that rare chromosomal anomalies are associated with a higher risk of congenital anomalies and adverse pregnancy outcomes. In this graphic, you can see the results of 4 interesting studies that describe the incidence and distribution of aneuploidies in different chromosomes. As you can see, there are adverse consequences for pregnancy related to almost all chromosomes. Another reason is that expanded NIPT can facilitate the detection of confined placental mosaicism when NIPT results indicate a rare anomaly which is not confirmed in the fetus. A placenta with a chromosomal alteration is not a normal placenta, it's not a healthy placenta, and for that reason can show different complications such as intrauterine growth restriction, miscarriage, perinatal death. In fact, according to scientific literature, the risk of intrauterine growth restriction and preterm birth, where NIPT has identified a rare trisomy, is from 29 to 47%. Another reason to not limit your screening to common trisomies is that expanded non-invasive prenatal testing can help to study possible reasons for pregnancy loss. Just very briefly, because Dr. Manuel will explain this point in more detail, to obtain a karyotype from conception products is sometimes challenging due to maternal contamination or cold tube failures, and expanded NIPT could be an easier and cheaper alternative. Okay, so now, Manuel, the presentation is all yours.
Okay, thank you, Alba. Now we are going to describe how our VeriRef platform works. In a pregnant woman's blood, you can find fragments of free DNA which come from the mother as well as the fetus in lower quantity. A blood sample is taken from the mother, the free DNA is sequenced, then processed on a computer, and a result is obtained. This is the work of the platform, from blood to report in just over 1 day. First step is using an automatic pipetter. Free DNA is extracted from the blood and the DNA library is prepared. The preparation of libraries consists of adapting fragments of free DNA that we have extracted before in such a way that the sequencer can work with them. The system is programmed to sequentially add primers, indexes, and adapters. Once the libraries have been prepared, they are taken to the massive parallel sequencer, which in our case is an exec 500 from Illumina. The sequencing is carried out using a flow cell. This is the flow cell, on the surface of which are placed some nucleotides that hybridize to the adapters added to the library. In this way, the DNA fragments are added to the surface of the flow cell and sequencing can begin. We perform 56 fluorescent PCR reaction cycles, and this way we obtain the sequence. In this way, we are able to obtain a 36 base sequence for every fragment. And now the computer is going to use the counting method. The informatics system will attempt to align with any chromosome the 35 base sequences. If it achieves to align with one and only one chromosome, the counting for this chromosome is increased. In this way, after roughly 3 million adjustments, a read number is established for each chromosome that we expect to be in a ratio for each chromosome, the proportion in relation to other chromosomes. Every chromosome has a proportion with the complete genome. If a chromosome, in this case 21, shows a higher proportion of that, we will have detected a trisomy. In this table, we can see how high to low severity are resulting effects of chromosomal alterations. In the trisomy of chromosome 1 and in any monosomy, except X, the embryo will not even be formed. In almost all the other trisomies, the embryo will not develop and the mother will miscarry in the first trimester. And so on until sexual chromosomes alterations with minor or no symptoms. In this table, we can see the prevalence of less common trisomies in different publications. The other great novelty of the VeriRef Gold test is the possibility of detecting deletions and duplications in all chromosomes. For this, the comparisons of signal intensity is used. When a signal decrease appears in an area, here or here, in relation to a normal sample, there is a delation, and the opposite is valid for a duplication. In this table, we can see the performance of VeriRef Gold, sensitivity and specificity near 100%. We are going to look at what NAPT cannot do. The first thing we should keep in mind is that although this is highly efficient, it's a screening test. So a positive test result must always be confirmed using an invasive test, ideally amniocentesis. Because there is a small possibility that the test result might not reflect the chromosomal status of the fetus, but might instead reflect chromosomal changes in placenta or in the mother, etc. Here we have an example of a condition that can confuse the test. Although the frequency that occurs is very low, the mosaicism confined to placenta is a cause of false result, since in the material blood, the fetal DNA that we find is mainly coming from the placenta. If the placenta not have the same composition as the fetus, it leads to error. When is VeriRef especially indicated? When older mother, high risk result in biochemical screening, echographic traces suggestive of chromosomal alteration, previous history of pregnancy with chromosomal alteration, couples wishing to discard chromosomal alteration, and as a first approach to assessing an early pregnancy losses. This is what mentioned Alba before. There is this recent publication by the group of Dr. Borrell, the Hospital Clínico of Barcelona. This publication establishes the usefulness of performing VeriRef Gold as an alternative to karyotyping of products of conception for the study of possible reasons for gestational losses. An algorithm for using the test for recurrent fetal losses is even suggested. Let's see how different alterations detected would be reported. In this first example, VeriRef Gold test detects a high risk of trisomy for chromosome 22. It is reported, and the client requests confirmation in amniotic fluid. The confirmation test is performed using macroarray, and the trisomy is confirmed. In the second example, VeriRef Gold detects a high risk for XW trisomy. it is reported, and the client requests confirmation in amniotic fluid. The confirmation test is performed by qF-PCR, and the trisomy is confirmed. And last example, VeriRef Gold detects high risk of copy number variant. Specifically, there is extra material from the short arm of chromosome 9. This is reported, and the client requests confirmation to an amniotic fluid tube. Confirmation test is performed by microarray, and the alteration is confirmed.
Okay, so now I will continue with some conclusions. Just to finish, I will summarize very briefly why we should not limit our screening to common trisomies. Fetal chromosomal anomalies are not limited to the most common trisomies, do not increase the positivity test rates in the average risk population, and are associated with an increased risk of congenital anomalies and adverse pregnancy outcomes. And also, expanded non-invasive prenatal testing can facilitate the detection of placental confined mosaicism and help to study possible reasons for pregnancy loss. And finally, it is very important to understand that it is a screening method not meant for diagnosis. How to order your VeriRef? Ordering VeriRef Gold is very easy. You just need to write to genetics@referencelaboratory.es and ask for your free VeriRef kit. You will then receive a kit with all the necessary documentation and struct tubes to collect a sample. Then the only thing you will need to do is to send back to reference laboratory the VeriRef box with the sample and its respective documentation. Once the sample will arrive to our laboratory, we will process it in 3-5 working days and share with you the medical report through RefLab portal. And to finish, I just want you to remind that we have an extensive portfolio of molecular genetic tests to meet each patient needs before pregnancy, during pregnancy, and after pregnancy. Thank you very much for attending today's session. We have enjoyed your presence. Okay, so we're going to have now a few minutes so that you can write any questions you might have in the chat. We will then have a look at them and answer them. Okay, we see that there are some questions, so in case we don't have time to answer them all, don't worry, those that are left will be answered by mail. Okay, so let's start answering those questions. This first one says, do you have a screening test for common trisomies? Okay, so yes, we do have it. It's called VeriRef, and it detects trisomy 21, 18, and 13. It also detects chromosomal aneuploidies and informs about the fetal gender.
Okay, so we have here another one. When is VeriRef basic test or VeriRef Gold recommended? Well, it depends on the expectations of the patient. VeriRef Gold covers a higher percentage of alteration, but has a slight higher possibility of false positive. Both tests are available, and it is the patient's choice to make a decision correctly advised.
Okay.
This one is for you, Alba?
Yes. So this one says, which is the turnaround time for your NIPT? Okay, our turnaround time is very short, just 3-5 working days, once the sample has arrived to reference laboratory. Okay, that's another.
This one. Yes, for me, how is the fetal fraction calculated? Well, the system uses the difference in size of the DNA fragments. the fragments of maternal origin are larger than those of fetal origin. The computer performs a distribution of fragments by size and calculates the percentage corresponding to fetal origin. This is the way.
Okay, very interesting. Okay, this question says, how must be the sample shipped? Well, you need to send 10 milliliters of maternal blood in a strep tube, and very important, all samples must be shipped immediately after sampling at room temperature, and at no stage they should be frozen, otherwise the sample could be destroyed.
Another one. How are the genetic alterations detected confirmed? Well, the confirmation depends of the alteration detected. Trisomies of chromosome 21, 18, and 30, and NPD for sex chromosomes are confirmed with QF-PCR. The rest of trisomies are confirmed with fluorescent in situ hybridation or microarray. And copy number variants always are confirmed with microarray.
Okay, so that's it. Thank you very much once again for attending. We sincerely hope to see you again at future events. Okay, so goodbye.
Goodbye, everybody.